Welcome to this lecture on the fundamentals of genetics. Today, we are going to explore a fascinating biological phenomenon known as a phenocopy. Have you ever wondered if your environment can change your physical traits so drastically that it looks like your actual DNA has mutated? We will discover how environmental factors can trick us into seeing genetic mutations that do not actually exist, and why understanding this is absolutely critical for doctors and geneticists. To begin our journey, let us start with a simple, everyday analogy. Imagine you were born with naturally dark brown hair, but you decide to visit a salon to bleach it and dye it platinum blonde. To anyone walking down the street, you look exactly like someone who was born with the genetic instructions for blonde hair. However, your actual DNA has not changed at all. Every single cell in your body still contains the genetic instructions for brown hair. If you were to have children, you would pass on the genes for brown hair, not the blonde hair you currently display. In genetics, when an outside environmental factor changes your appearance to perfectly mimic a genetic trait you do not actually possess, we call this a phenocopy. Now that we understand the basic idea, let us look at the formal scientific definition. A phenocopy is defined as an environmentally induced, non-heritable variation in an organism's physical traits that closely resembles a trait known to be caused by a specific genetic mutation. The crucial point here is that the organism exhibiting the phenocopy possesses a completely normal, wild-type genotype for that specific trait. Its DNA is perfectly healthy. The altered appearance is strictly the result of exposure to specific environmental conditions, such as extreme temperature shocks, chemical agents, or nutritional deficiencies during a critical period of development. Because the underlying DNA sequence remains completely unchanged, this induced trait cannot be passed down to the organism's offspring. It acts as an environmental imposter. Understanding this concept of an environmental imposter brings us to its real-world importance. Why does distinguishing a phenocopy matter so much? It is absolutely critical in the fields of medical genetics and genetic counseling. When a doctor sees a patient with a specific physical abnormality, they must carefully determine whether it is caused by a hereditary genetic mutation or an environmental factor. If the condition is a true genetic mutation, the patient has a specific, calculable probability of passing the disease to their children, requiring extensive genetic counseling. However, if the condition is merely a phenocopy caused by an environmental exposure during pregnancy, the genetic risk to future children is essentially zero. Without this knowledge, doctors might incorrectly diagnose an environmental injury as a heritable disease, causing unnecessary panic. To see how doctors and scientists analyze this formally, let us look at the mathematics behind physical traits. We can understand phenocopies mathematically by looking at the equation for phenotypic variance. This formula describes the total variation we see in a physical trait within a population. VP represents the total phenotypic variance, which is the observable physical differences among individuals. VG represents the genetic variance caused by actual differences in their DNA. VE represents the environmental variance caused by outside factors like diet or chemicals. Finally, VGE represents the variance from gene-environment interactions. In the case of a perfect phenocopy, the genetic variance for the mutant trait is exactly zero. The entire shift in the observable trait is driven by a massive increase in the environmental variance. Now that we have our variance equation, let us apply it to a tragic but important historical example. In the late 1950s, many pregnant women took a drug called thalidomide to treat morning sickness. thousands of their babies were born with a severe condition called phocomelia, where limbs are extremely shortened. This perfectly mimics a rare recessive genetic mutation called Roberts syndrome. We are asked to evaluate the source of this trait. Because the babies do not actually have the mutated genes for Roberts syndrome, the genetic variance is exactly 0. The gene-environment interaction is also 0. Substituting these into our formula, we find that the total phenotypic variance equals the environmental variance. The final answer shows the trait is purely driven by the chemical environment in the womb. Having calculated that the variance is entirely environmental, let us prove logically what this means for the next generation. This chunk of our analysis establishes the non-heritable nature of the condition. Because the genetic variance is 0, these children do not have the mutated genes to pass on. The mathematical reasoning tells us that if the environmental trigger is removed, the phenotype must return to normal. And indeed, when these affected children grew up and had their own babies without taking thalidomide, their children were born with perfectly normal limbs. This proves conclusively that the thalidomide-induced phocomelia was a classic phenocopy. It acted as a perfect environmental mimic of a genetic disease, but the underlying DNA was always healthy. Beyond population variance, we can also look at this on an individual level. This brings us to another simple but powerful mathematical representation. We can define the relationship between an organism's physical appearance, its genes, and its surroundings using a simple function. P equals a function of G and E. Here, P represents the observable phenotype, or what the organism looks like. G represents the genotype, which is the actual genetic makeup, or DNA sequence. E represents the environmental conditions the organism is exposed to during development. If the environment E changes drastically, the phenotype P can change to look like a mutation, even if the genotype G remains completely normal and unchanged. Let us see how this works in a laboratory setting. Let us apply this function to a famous laboratory experiment. In fruit flies, there is a known genetic mutation called cross-veinless, where flies lack tiny veins in their wings. A researcher takes normal, wild-type fly pupae and exposes them to a brief, intense heat shock. We are asked to determine the resulting phenotype. We know the genotype G is normal, which we will call capital C, capital C. The environment E is extreme heat at 40 degrees Celsius. When we plug these into our function, the extreme temperature disrupts protein folding during a critical developmental window. The final answer is that the resulting phenotype P is the cross-veinless appearance. The heat shock has created a physical trait that perfectly mimics the genetic mutation. Now that we have seen the flies develop the mutant appearance, let us prove what happens to their offspring. This step is crucial to confirm it is a phenocopy. The mathematical reasoning here relies on the fact that the genetic component, G, remains completely normal. The heat shock did not alter the DNA. Therefore, the offspring will inherit the normal alleles. When those offspring are raised in a normal environment, where E equals 25 degrees Celsius, we evaluate our function again. With a normal genotype and a normal environment, the phenotype P must return to normal. The offspring will have wild-type wings with intact crossveins. This proves that the cross-veinless appearance in the parents was purely an environmental imposter. To solidify everything we have proven so far, let us look at this flowchart summarizing the mechanism of a phenocopy. On the top pathway, you see the standard biological process. A normal genotype paired with a normal environment leads to a normal phenotype. In the middle pathway, A mutant genotype naturally produces a mutant phenotype, even in a normal environment. But the bottom pathway is where the magic happens. Here, we start with a completely normal genotype, but introduce an extreme environment, like chemicals or temperature shocks. During development, this forces the organism to produce a mutant phenotype. As the curved arrow indicates, this bottom result perfectly mimics the genetic mutation from the middle pathway, despite the DNA being completely healthy. We have covered a lot of ground today, so let us summarize the most important ideas. First, a phenocopy is a fascinating biological phenomenon where an environmental factor forces an organism to develop a physical trait that perfectly mimics a genetic mutation. Second, and most importantly, the organism's actual DNA remains completely unchanged throughout this process. Because the underlying genes are not altered, these environmentally induced traits are never passed down to the next generation. Finally, understanding this concept allows scientists and doctors to accurately distinguish between heritable genetic disorders and non-heritable environmental injuries. You now have a solid understanding of how the environment can act as a genetic imposter. Great job following along with these fundamental concepts of genetics.